Good morning, students. Yeah, am I audible?
Yes, ma'am, you're audible.
So I was thinking that some of you might have opted for offline classes, but I could not see any faces from BSc semester 5 here. So that means all of you have opted for the online classes, right? Okay, so 10 students have joined and few are still to join. Let's wait for 2 minutes more and then we can start the class. Okay, so let's start the class. I think still many students are not here. Was there some or other class going on, some exam going on, Manaswani?
No, ma'am, there was no exam. But our class ended at I think like 10, 12 or something.
Okay, fine,
There was no.
fine. Thank you. So yesterday we discussed about the lab safety and we discussed that why lab safety is important, what is lab safety all about, everything. So let's quickly just revise some of the topics that we discussed yesterday. So there was one term that we used, hazardous substance and a biohazard. So what is difference between hazardous substance and a biohazardous? Yes, Ishita, can you answer this question? Hazardous and biohazardous. What is the difference?
Yes, ma'am. Ma'am, am I
Yes.
audible?
Yeah, you're audible. Please switch on your camera also so that I can see you.
Okay, ma'am. Just a minute. Ma'am, am I visible?
Ishita, I guess not now, but okay, your camera seems to be on. Please answer the question.
Ma'am, hazardous substances are those substances which are which are dangerous to us.
Mm-hmm.
And biohazardous substances are those substances. Ma'am, am I audible?
Yeah, you're audible, please.
Biohazardous substances are those substances which are specifically dangerous to each healthy living organisms. Basically, which they are dangerous to the health of a living organism.
Okay, yes, Devanshi, correct Ishita where she is missing the point. Yes, Devanshi. Devanshi, where are you? You are suddenly offline. Okay. Yes, Manaswani, correct her.
Ma'am, so hazardous substances can be any substance which is harmful to another individual. So, for example, it can
Mm-hmm.
be any kind of chemical or products like that. A biohazardous substance is again it is a hazardous substance, but then it has some kind of biological origin. So it could be any microbe or some kind of pathogen or anything else that we're working with, which has
Mhm.
some biological origin, which can be harmful to us.
Yeah, that's right, Manaswani. So yesterday there was another term that we used very commonly. It was used, that was the containment. So what does containment means? Yes, Janvi, can you answer this question? What does containment means? We had a very long discussion on this particular word yesterday, right? Those students who are not joining on time, next time से मैं late turn ons off कर दूंगी, and after 5 minutes students class ही नहीं join कर पाएंगे. So please inform everyone that at least by 10:20 you should be here in the class. Otherwise I'm not going to allow you to enter my class. Right? Yes, Nehal, answer.
Ma'am, containment means that we keep something harmful under control to prevent the
Yeah, under
infection.
control.
Yeah, ma'am.
Yeah, so that it's not accessible to everyone, so that if it is a pathogen, it is not, people are not get affected and it does not spread, right? So that is the containment. Okay, so who will tell me that what is, why every kind of a chemicals which are present in the lab, or the different kind of the buffers that we prepare, they should be labeled? Why labeling is important? Namrita? Yes, you, Raj, you can answer this question. I can't hear anything, Yuvraj. There is a lot of background noise and not even a single word is audible to me.
Am I audible now?
Yeah, you're audible now.
So labeling is important just because to have some idea about what the chemical is all about and its characteristics
Mm-hmm.
and what harmful effect it can show and what measures we can take while using it.
Okay, fine. Okay, now who is going to tell me what are MSDS? Expand the term MSDS and why they are useful. What kind of data they have. Yes, Sahiba, can you answer? Nandini, can you answer this question? Yes, Shakti, you can answer.
Yes, ma'am. So MSDS is referred to as material safety data sheets. So they provide information about the chemicals, their uses, and the precautions we need to take. And also, if something happens, so they also provide the information about the first aid we can use.
Yes, that's right. That's right, Shakti. So what is PPE? Who is going to tell me what is PPE? Kshitij, what is PPE? Only few students always raise their hand. Only few students always answer in the class. Rest all what they exactly are doing, I don't know. Yes, Sam.
Ma'am, PP stands for Personal Protective Equipment, and it's used to protect yourself from hazardous and biohazardous materials.
Okay, yes, that's right. PPE is a personal protective equipment. So when you are working in the lab, it helps, it basically prevent our direct contact with the chemicals or the pathogens or any other thing that we are using in the lab. So basically it is a kind of a barrier. It is a kind of a barrier that helps us in a many ways. So well, when we are dealing with the, sometimes with the pathogenic microorganisms also, or the viruses or pathogens where transmission rate is very high, so different kind of a PPE are being recommended according to the pathogen that we are dealing, or according to the research work that we are doing, right? So okay, we discussed this, and today is again the continuation of the lab safety, and we are going to discuss about uh, the risk groups and biosafety levels majorly, that what are risk groups and what are 3 biosafety levels or 4 biosafety levels, what kind of a different facilities are there in the 3 levels, and how at each level they're increasing, in sensitivity also, like they are more sensitive as we are going to the higher biosafety levels, right? And how we are handling different pathogens. So just before that, some other terms are like sharp. Like sharp is any kind of any item basically which has acute and rigid edges and the corners which can cut, which are capable of piercing, ripping, or puncturing, such as syringes, blades, and broken glasses. So, all these kinds of things are labeled in a sharp. They are labeled as a sharps, and when there are certain practices that when we use these kind of a sharp thing, there are always risk of being cut or being pierced by these kind of a thing. So, if we use hypodermic needles, then we need to be very careful and discard. Their discard is basically very important, that they are being discarded in a closed container so that they do not cut or they do not protrude in other persons also, or persons who are handling the waste material. So a separate container there, and that container is labeled with the sharp, so that the person who is handling that kind of a waste material also be cautious that they can get harmed by these kind of a material. So all sharps, they must be placed in a rigid, puncture, and a leak resistant container, and which is also impervious to the moisture. So sharp container must be labeled either with the biohazard or infectious phase, depending upon the kind. Like for example, hypodermic needles sometimes are used to take out the blood from the animal if some kind of a testing is being done. So they could be infectious because the animal that we are dealing may be artificially inoculated with some pathogen that we are studying or some other thing. So in this way, they should not come in a direct contact with the people, those who are handling those kind of a waste. Now another important thing, when we talk about the lab safety and biosafety, is the risk group. So what is risk group basically? See, whenever we are doing any kind of a research, there's always some sort of a risk associated with that kind of a work, right? So when we are working with the pathogens, when we are working with the even if we are not working with the pathogen, we are doing some kind of a research in the lab. So there is also some type of always some type of a research which is being associated with that. So how we categorize them. So this is the targeting the lab. Basically, risk group is basically for those laboratories that work with the infectious agents, right? So basically, it is determined by the kind of the research that we are doing. So if there's a more hazard with the agent or a material or the pathogen that we are dealing with or on which we are doing research, then the higher the risk group, right? So therefore, those kind of pathogens, if we are dealing with the higher risk group pathogens, then there should be more stringent and containment practices that we need to follow. As if risk group is less, so we can be okay, BSL level 1 can be followed for the risk group 1 kind of pathogen, where the stringent guidelines or containment facilities are not there, so normal lab can be done with. So biological agents are basically, they are being categorized in the risk groups. It is just directly proportion to the kind of the risk they are causing to the people who are coming in direct contact with these kind of pathogens, right? So basically, how they are being on what criteria these pathogens are being categorized into the different risk groups. First is the pathogenicity of the organism. Pathogenicity means ability to cause the infection, how much infectious that pathogen is. Mode of transmission and host range, how that pathogen is being transmitted. Are there some kind of a vectors that it require, or it is being transmitted through the droplets or fomites or aerosol generation? What kind of a transmission mode is required, right? And the host range, number of the people or the number of the plants or humans. Basically, risk groups, we don't categorize the plant pathogens in the risk group because we know the plant pathogens are not going to infect us, right? So basically, host range means the people. Our particular age group of people are more prone to this kind of a pathogen, or they can impact anyone in the population. So, and then availability of effective preventive measures, that is for when we talk about the pathogens or viruses, especially the vaccines, the kind of the pathogen that we are dealing with, is this already studied pathogen? So if it is already studied pathogen, so there must be some therapeutics or vaccines which should be available, or this is some new kind of a pathogen that we are working with and there are no vaccines available, right? So on that basis also, it is, this point is being carefully studied. Then availability of the effective treatments, if are there any antibiotics which are available or not, and some of these factors which are majorly being studied very carefully when we designate to study particular pathogen under a different risk group, and then when we study them in the different biosafety lab or BSL facilities. Then potential outcome of the exposure, if someone get exposed to that pathogen while working, what could be the outcome? क्या वो person भी infect हो सकता है? Is there any possibility? If yes, then what will be our next step? Natural route of infection, naturally how this infection is being spreading and how it is being done. Other routes of infection, maybe that could be due to the lab manipulations. Is it airborne or ingestion, or what kind of a, like, and then the stability of the agent in the environment, how much stable this agent is in the environmental. Then concentration of the agent and volume of the concentrated material to be manipulated, in what volume we are going to study that pathogen, presence of a suitable host is there or not, information available from the earlier studies and reports of lab acquired infections or any other clinical reports if they are available, and local availability of effective prophylaxis or therapeutic interventions. So are there any interventions directly available for this pathogen or not? So we take into consideration all these factors, and then these are being designated into risk group 1, 2, 3, and 4. So risk group 1, agents which are not associated with the disease in healthy adult humans. That means they have a very low individual as well as very low community risk. For example, Bacillus subtilis or E. coli strains, like we can go and work with them in the normal BSL-1 facilities. BSL-1 facilities usually is normal lab facilities which has a laminar A-flow hood which is available. So it is not, we don't need a special kind of a lab or a particular design. Just a laminar flow is there, and we can work in that laminar flow air hood where the HEPA filters are being properly as per the requirement are in the laminar flow hood available, right? Then risk group 2. So these are the agents or pathogens which are associated with the human disease, which is really very serious. And if they, this for these this kind of a diseases, preventive and therapeutic interventions are available. That means it has a moderate individual risk. Yes, it can infect the person who is working with that pathogen, but then we have already therapeutic interventions which are available. So it is not very risky, and it has a very limited community risk. Then we example of such kind of pathogens are Streptococcus aureus, which causes minor skin infections. E. coli causing severe diarrhea and death, antibiotic treatment is available. Then Bordetella pertussis causing whooping cough will be associated with death, antibiotic treatment and vaccination is available in these. So agents basically here, they can be very serious or they can cause lethal human diseases, and there are chances that for these diseases there may be a preventive or therapeutic interventions available and may not be. Certain chances are that there might be available, but then it could be certain probabilities there that they might not be available. So they have a high individual risk, but still the community risk at this level is very low. Then Mycobacterium tuberculosis causing tuberculosis. It is there. It has a lengthy antibiotic treatment. We still don't have a vaccination, a proper vaccination, reliable vaccination, we can say, once this disease happen. It is not there. Then SARS-CoV-1 and MERS-CoV virus causing respiratory syndrome with a high mortality can be treated here. No vaccination and no effective treatment is available in this case, but still at risk group 3, they are placed in risk group 3 and can be manipulated in BSL lab 3 facility. Now risk group 4 is the highest, at the highest level. That means here we study the pathogens where we don't have any preventive or therapeutic interventions. Usually we don't have any, and they are very serious, and their transmission rate is very high of these kind of a pathogen. For example, the Ebola virus. The high risk SARS-CoV-2 virus also poses, it is a high community risks, right? So if we want to do any kind of a manipulation on this virus also, we need to go for a BSL level 4 facility, right? So at this time, SARS-CoV-2 virus is also being studied at the BSL level 4. Then Ebola virus, it causes hemorrhagic fever, is again associated with the high mortality rate, while the vaccine is available, but then still not worldwide, and very effective treatment is not available for this virus also, right? So once we know the risk group of a particular pathogen, once see, for example, we want to work on a particular pathogen, then first thing we study is that under which risk group this pathogen is being categorized. So after it is being categorized in a particular risk group, so corresponding to that, we'll know that in which biosafety level we need to work on this pathogen, right? So along with the risk group, there is associated biosafety level. And this is same principle as risk group, is that as the risk increases, the containment and practices, they become more stringent. For most cases, our lab is the risk group 1, then BSL level 1 is like we have a labs in the university here. So we have a BSL 2 also, but most of the lab in the university levels, even if some kind of a research is being done, they are BSL 1 facilities. So different BSL biosafety levels has been established, and preventative measures against the human infection. Why we have established these biosafety levels? So that we don't get direct exposure to these pathogens, and this somewhere prevent the infection in the persons who are being working continuously on these kind of a pathogens. So taking precautionary measures can help to minimize hazards which are associated with the infectious agents. As the risk increases, a biosafety level also increases, and each biosafety level has a specific containment practices also. So at the lowest of the 4 is the BSL level 1. So it is normal lab setting where we work with the low risk microbes, and these kind of a microbes, they have a very little or no risk to the humans. And example is, for example, non-pathogenic strain of E. coli is very commonly used, and being very commonly used in the microbiology experiments also, and we do all kind of experiment in the BSL lab 1. So this kind of the lab setting consists of a research taking place on a benches without the use of any containment equipment. So this lab is also not required to be separated from the surrounding facilities. It can be just among other lab works that we are doing. We don't need a separate facility. So again, here also certain guidelines are being provided, such as mechanical pipetting only, mouth pipetting is not recommended. Then avoidance of splashes and aerosols, daily decontamination of all work surfaces when work is complete, hand washing. So normal good lab practices or a good microbiological techniques we follow, wearing a personal protective equipment, then biohazardous signs if required. So BSL lab ones also require immediate decontamination after spills, and infection materials are to be decontaminated prior to disposal. So this means, so normal labs also after certain period of a time, they are being decontaminated. There are some kind of a decontamination practices which are being done, so that usually we work with a different kind of pathogens in the lab. Sometimes the material in the washing or decontaminated material, or even not, before we do it decontamination, we keep those kind of a material on a certain kind of a bench. So somewhere these kind of a pathogen can be found there in the lab. So the decontamination for the lab after a certain period of a time is also being recommended. Then BSL level 2, then these covers, or these kind of a labs, they deals with the pathogens which causes human diseases, and they cause a moderate health hazard. For example, equine encephalitis viruses, HIV, Staphylococcus aureus, these kind of a pathogens, they can be easily handled in the biosafety level 2 facility. They maintain the same standard practices, the formal guidelines to work in a lab in a BSL 1, they are being followed at the BSL 2 also. Then again, then people need to be more careful to prevent injuries such as cuts and other breaches of the skin or ingestion or a mucous membrane exposure in this level. And in addition to BSL 1 expectation, the following practices are required in BSL Lab 2. Appropriate personal protective equipment must be worn, including lab coats and gloves. Eye protection and face shields can be worn if required. Uh, any procedures that we are doing, if they can cause infection from aerosols or splashes, they are performed within the biological safety cabinet. So these cabinets are designed in such a way that the air which is inside the cabinet is not being circulated towards us. It's just circulated inside the cabinet, and it is taken outside that particular area by the uses of certain HEPA filters are there and certain exhaust fans. So they take that air which is in the environment in the that particular biosafety cabinet directly to the outside. It doesn't come inside, right, or towards the person who is working. So they can work in that particular area. So a procedure, then again, autoclave or alternative method of decontamination is available. Autoclave, we know, is important method for sterilization, right? So for sterilizing many things, media and other things, we go for autoclaving. Similarly, for decontamination also, autoclaving plays a very important part. So autoclave, which is only being used for the decontamination purposes in BSL Lab 2 facility, is being kept there in the facility. So you don't need to go out and take out these facilities, or maybe for decontamination, you don't need to go out and do the decontamination and then use other facilities. Then lab has self-closing lockable doors. There's a sink and an eye wash station where available. Biohazard warning signs are always there. Then access to such lab is more restrictive than BSL 1 lab. So only people who are working in that particular area are being allowed to go. So outside people, especially those with increased risk of contaminations, are restricted from entering when work is being conducted. So BSL 2 lab is also not open for everyone. Only people who are working, who need to work in the BSL lab facility, they will go in that facility, and other peoples are not allowed to enter those facilities. Then we have a BSL 3. Again, building upon the prior biosafety level, BSL 3 lab includes work on microbes which are either indigenous or exotic and can cause serious or potentially lethal diseases through inhalation. So, for example, yellow fever, West Nile virus, and bacteria which causes tuberculosis. Some bacterial strain which causes tuberculosis are also being dealt in the BSL level 3. So microbes are so serious that work is often strictly controlled and registered with appropriate government agencies. Lab personnel are also under medical surveillance and could receive immunizations for microbes they work with. So health of the people who are working in this kind of a lab is important. So before they are being allowed to work in this kind of a lab, there's a proper health checkup, and if they are required for a vaccination or immunizations for the microbes they are working in, so that is also being done. BSL 2 lab design, if you talk about the design feature, ample space must be provided for the safe conduct of lab work and cleaning and maintenance. Walls, ceiling, and floors should be smooth, easy to clean, corrosive resistant, or chemicals and disinfectants can be used there. Bench tops should be impervious to water and resistant to disinfectants so that they can be easily cleaned. Illumination should be adequate. This is very important. Then storage space must be there so that you can put your things inside. Additional long-term storage space conveniently located outside the lab working area should also be provided. Then important things, facility for eating and drinking and rest should be provided outside the lab working area. Hand washing basins with running water should be provided near the exit door. Then doors, they should have a vision panels, appropriate fire ratings, and self-closing. Then at BSL level 2, an autoclave or other means of a decontamination should be available. Then first aid rooms should be suitably equipped, right, and easily accessible. Waste handling. Now, how to handle waste which is being generated in these kind of a lab. So waste is anything which basically is discarded. So it's not that whatever waste is being generated and we go out and discard it. No, the waste is being segregated, it is being decontaminated, and then it is being properly discarded. So decontamination of waste and the ultimate disposals are very important. In terms of daily use, if any contaminated material will require actual removal from the laboratory or destruction, then most glassware instruments and lab clothing will be definitely reused and recycled. We don't discard them. They are being washed properly and they are being sterilized and reused, right? So the main objective is that all infectious materials should be decontaminated, autoclaved, or incinerated within the laboratory. So this is how it should be disposed. So decontamination, steam autoclaving is preferred method for all decontamination processes. Then they should be properly placed in a proper containers and then decontaminated, right? And then BSL-3. So common requirements in the BSL-3 are a standard personal protective equipment must be worn. All work with microbes must be performed within appropriate levels. Access, hands, free sink, and eye wash should be available in the exit. Then sustain directional airflow to draw air to the lab from the clean areas toward the potentially contaminated area, right? That means exhaust air should not be recirculated in any case. A self-closing set of locking doors, and again the access to this kind of a laboratory restricted and controlled at all the time. So this kind of a labs are being usually not built in the area where the movement of the people is more, usually different kind of a buildings or away from the places where the most people are present. So in isolated places they are being recommended. So designs and facilities are similar than the in which are being in the 1 and 2. I don't think there is any difference. They have the similar kind of a thing, so you can go through that. Okay, they have an enter room, right? So enter room should have a facility for separate cleaning and dirty clothing and a shower if required. Then again, their rooms may be self-closing and interlocking, and a breakthrough panel may be provided for emergency exit use. Again, the surface wall and ceilings should be water resistance as in the earlier cases. Windows must be closed, hand washing station should be there, so it is similar. Then the exhaust air from class 1 or class 2 biological safety cabinet which have been passed through HEPA filter must be discharged in such a way to avoid the interference with the air balance of the cabinet or the building exhaust system. So there's a proper construction of the BSL Lab 3 facilities being done by the specialized people. So how they design it, how they make the exit of these exit pipes so that the airs, they are drawn out, directly outside through the atmosphere and to a level, to which higher level. So that is being recommended, that is also being done. Then autoclave for decontamination is used here also. Then okay, here the medical examination, all lab personnel who work in the biosafety level 3 and up is important, right? So detailed medical history, and thorough medical examination, and after then only they should be employed. Then highest level of a biosafety is the BSL-4. So the pathogens, usually the pathogens for which no therapeutics are there, or some newly identified strains of the viruses or new pathogens are being studied in BSL level 4. They have following containment. What's wrong with this? Okay, so personals are required to change clothing before entering, and there's a shower upon exiting. So shower is mandatory. Decontamination of all material before exiting. They must wear appropriate PPE, again full body, air supplied, positive pressure suit should be there. Then it is a extremely isolated building, often located in a separate building or in an isolated or restricted zone of the building. Then okay, this is the same. Decontamination, all effluents from the suit area where they work or a decontamination chamber, contamination shower must be decontaminated before the final discharge. Heat treatment is a preferred method. Effluents may be required to correction to a neutral pH before discharge. So these requirements are there. Again, the water from the personal shower and toilet may be discharged directly to the sanitary sewer without treatment. Then sterilization of a waste material is important. A double door pass-through autoclave should be available. Then other method of a decontamination should be available for equipment also. And emergency power and dedicated power supply should be there. Containment drains should be installed. So this is a comparative way. This table basically shows the BSL1, BSL2, BSL3, and BSL4 facilities, how which kind of a pathogens we deal, pathogen type, and what are the autoclave requirement in the different biosafety levels. How at different biosafety level, in which biosafety level we need containment. For example, if we talk about containment, BSL level, it does not require any containment, and BSL level 2 containment, but yes, still not that high level. At BSL level 3, high containment, BSL level 4, maximum containment, right? So E. coli can be, we can deal with the E. coli in BSL level 1, influenza, HIV in BSL level 2, tuberculosis 3, BSL level 4 may Ebola viruses or other pathogen which causes maximum human hazard and human diseases, or their transmission rate is very high. So biosafety and lab safety plays a very important role when we are working in any kind of lab, starting from a simple normal lab to the high-end lab where the research is being done. So biosafety level 4 are not installed in the private laboratories. These are always under some kind of a government agencies or a government organizations. They are part of this, like they are not individually being offered to the labs to work to be done, right? So they are being taken under the direct control of the government. They are responsible for it because they are maximum highly infectious pathogen are being handled at this level. So there is a maximum risk to the people, or a direct risk to the people who are handling these kind of a pathogen. So a lot of precautions and a lot of handling care is required to be taken. So in BSL level 4, the training of the personals, training of the people who will work under in such facilities is also required, that they should be trained properly about how they should work in the lab. And there's a two people policy in when we work in a BSL level 3 and a BSL level 4, that is no individual person will go alone in such kind of a lab. There should be always one person accompanying, so that in any case, in any mishap or in any cases, those accidents can be reported to the outside properly. So these are the guidelines which are being followed in the for the biosafety labs or different labs in a normal labs that we work. So what are the precautions that we require to take? So if there's anything that you need to ask, please ask your questions. In the meanwhile, I'm going for one. This time we'll not go for any written question. We'll do one discussion on a simple topic today, right? 10-minute discussion. So if there's anything you want to ask, please ask. Okay, that's very good that you understood everything. So I think, yes, Manaswini.
Ma'am, so would the coronavirus come under BSL 4 or BSL 3?
See, I'm telling you, see, when we are doing normal testing and normal other things, sample preparation can be done in BSL 3. But if we are using going for the autopsies of the people and using larger tissues for the isolation of the virus, then that will be done at the level 4 only. Otherwise, for normal diagnostics and other things can be done at the level 3. Sample preparation can be done at the level 3 only.
Okay, okay, ma'am. Thank you.
Okay, so just a moment. Okay, so this basically finish the first module, this topic about the lab safety. So I don't want you to all to mug up all the things. Just try to like in this last table, there's a comparative way where we are talking about the different kind of a BSL lab. So a comparative way where the risk group are directly linked to the BSL Lab 4. A comparative, you can make yourself a table where you can enlist the properties of a BSL 1, 2, 3, and what are absent in 1 and what are present in a 3 and 4. So that way it will be easy for you to remember that, because most of the requirements will be same, except that at each level something new will be added, right? Like shower is being added after 3, health checkup is added after 3, right? The containment becomes more stringent after 3. So a comparative way of a learning would be easy for you to remember the things, that what are the BSL3, what are, how we are handling, and how the stringency level is increasing at each level, right? Okay, so that's this is, and okay, I said that we'll have a one discussion for different kind one on this topic basically, right? So okay, let's see uh, what kind of other pathogens can be, like other than I discussed today's lecture, can be we can deal at BSL level 1. Can you name some pathogens which can we can deal at BSL level 1? अपने लैब में कुछ प्रैक्टिकल्स किए होंगे कभी, किस किस को आप हैंडल कर सकते हो?
Streptomyces.
Streptomyces, yes.
Lactobacillus.
Lactobacillus, bacillus, yes.
E. coli.
Yes, E. coli is very commonly, and we use this for. Okay, yes. Oh, what about BSL level 2 then? What kind of a pathogens we can deal at BSL level 2 then?
Ma'am, Streptococcus pneumoniae.
Mm-hmm.
And
HIV. What about BSL level 3? Okay, so fine. Let's do it this way. So just all of you just prepare a list about of at least 5 minimum pathogens that can be we can study at each level, and that list we'll discuss in the next class on Friday after the practical, right? So just prepare one list where of 5 pathogens which we can deal in level 1, level 2, level 3, and level 4, right? That way it will be more easy. So we can discuss this list in the second or next class that we have, right? So for your attendance, all of you, please switch on your cameras. Please just do switch on your cameras at least while I take your attendance, because it is mandatory, right? So yes, all of you now to all are present. Many students, they did not join the class even after I guess there was a 10:25 also, right? So please make sure that by 10:20 you all join the class. Otherwise मैं late turn ons आपका joining off कर दूंगी, and then you will not be able to join the class. Make sure that you all join the class in time. 5 minutes are more than enough to shift from one class to the other class, right? Yes, Adish Mishra.
Present, ma'am.
Athoiba. All of you please switch on your camera. Athoiba.
Present, ma'am.
Ayushi, yes, Athoiba, I can see you. I can see Adhish, I can see Ayushi. Sam, I can see you. Yes, Sam. Devanshi. Devanshi, you're muted, please.
Present, ma'am.
Okay, Devanshi, you're there. Then Ishita Arora is there.
Present.
Yes. Janvi. Kshitij. Manaswani, yes, Manaswani is there. Namrita.
Present,
Yes,
ma'am.
Namrita is there. Nandini.
Present, ma'am.
Yes, Nandini is there. Nehal Joshi.
Present, ma'am.
Nehal is there. Rupal is there. Sahiba.
Present, ma'am.
Yeah, Sahiba is there. Sanjana Sharma. Yes, Sanjana is there.
Present, ma'am.
Shakti.
Present, ma'am.
शक्ति के बाद किसका नाम है? शिवानी मिश्रा. Shivani is there.
Present,
Tanvi
ma'am.
Sharma. Yeah, Tanvi, Tridev,
Present, ma'am.
is the list on. त्रिदेव के बाद है युक्ता, विवेक and Yuvraj. Yes, Yuvraj. Yeah. Okay. Fine. So I'll see you day after tomorrow. We don't have a class tomorrow, right? So day after tomorrow, I'll see you and we'll start with some new practical, right? Okay. Thank you so much. Yes, Shakti.
Yes, ma'am. I have a question.
Mm-hmm.
Ma'am, how can we reduce the effect of radiations if we are like working on radiations? So can we reduce its effect on us?
Yes, it can be reused. There are certain equipments which come such as lab coat. So इनका जो lab coat होता है ना, वो किसी I don't remember asbestos से या किसी चीज से prepare होता है. It is very thick and very heavy. I don't know if you all have visited sometime ultrasound on those radiation labs. Those people are always wearing that coat. So that coat prevent the radiation penetration. अब मुझे उसका material याद नहीं है, next class में बताऊंगी. It's very thick, very heavy. I don't remember वो किस चीज का बना होता है, but yes, it can be prevented. And there are face shields are there, are proper shields which the radiations will not penetrate. And then the other things are that when we are working with the radiation, there is a scintillation counter, one equipment which is present in the lab. So that counter beeps when it senses the radiation more in that particular area. So these kind of equipments and instrumentations are being placed in the environment or a lab when we are working, and they definitely helps and they prevent our direct exposure to the radiation. But we have a glass shields when we work, and there's a proper face shield, a proper very big face shields like a helmet thing that we use, and a very heavy coat. उसका मुझे material बिल्कुल याद नहीं आ रहा है अभी किससे बना होता है. And when I was doing my PhD, we used to do a lot of hybridization. So I used to wear that coat, and after wearing that coat for 2 days, my back was like this, and I was not able to walk straight because it was very heavy. So I'll just उसका नाम next class में I'll just tell you, right? Okay.
Thank you, ma'am.
Okay बच्चों, I'll see you then day after tomorrow. Thank you so much.
Thank you, ma'am.
Thank you, ma'am.
Thank you.